with Chesney Michaels at Unum to talk about another step in the manufacturing process of T cells. So this originally was a blood cleaner, but
Yes.
now you're going to use it to actually separate cells for therapeutic use. So what's the difference and how does this work?
So the way the machine works is very similar to a centrifuge.
Okay.
So it's a centrifuge that allows you to pellet cells in a specific way, pellet cells based on a speed that you need to do. And then at the same time, so it concentrates the cells. So pellet cells meaning I'm concentrating my cells of interest, but at the same time I might want to clean that cell so I can wash that. So why this machine is beneficial for us in a PD or in a manufacturing setting is it can act as another machine or system to allow us to either concentrate cells or wash that cells to do a media exchange. So in that setting, that's where we are using this machine.
So in order to understand what this machine is going to do, I see you've got some bags up here. What are these bags representing in the cell therapy manufacturing setting?
So in the cell therapy manufacturing, first of all, I want to say that the machine itself can act as a backup to the prodigy.
Okay.
So in the scenario, we're going to use this machine as an alternative to do Ficoll. Ficoll to separate the cells, either to separate the PBMCs from the rest of the red blood cells and the granulocytes.
Okay.
So in these bags, what we have here is, if this was in a particular setting that we would want it in, this would be your leukapheresis pack coming from the patient. Over here you have a buffer. And then on this side, we have the Ficoll, which will allow us to separate the cells.
Okay, so this is a material that has a specific density that allows us to do density-based separations.
Yes, under centrifugation.
Under centrifugation. But instead of being in a falcon tube, which is where I've seen it before, we've got it in a bag because it flows through the machine.
Yeah. And
Okay.
the bag itself is a closed system, unlike a falcon tube, which you would have to open, or you might have to be in a hood. In the bag, it's closed, so as you can see, we're standing in an open space, so everything inside should be sterile.
Okay. And under here, this looks like the centrifuge component?
Yes, so this is a chamber, what's called a bowl in terms of this particular machine. And these bowls come in 3 sizes. They can either be 70 mils, 125, or 225.
Okay.
So that depends on what you would like to use it for. Would you just like to wash the cells and you need a lower volume? Or based on your concentration of cells, like what concentration you want to concentrate the cells to? Or in this case, for Ficoll, we use the biggest bowl.
Okay, so now that you've got everything set up, why don't we check the bags down at the bottom?
Yeah, so one of the things you want to do is you want to make sure that all your clamps are sealed because you want to be able to control the path of the liquid flowing.
Okay.
So, and you also want to make sure that, as I said, you want to control the path of flowing and that the right paths are open so that it doesn't generate a pressure alarm and you don't have the machine shouting at you.
Okay.
So on the side of this machine, we have a bag that will collect some air. So right now there's air inside that bowl.
Okay.
So that air will get pushed out into this bag. And this is how the machine, one of the ways the machine controls the volume, the movement of the volume. It uses the air to then move the volume in either forward or back, depending on where you open the clamps for the path.
Okay, so the air is the working fluid that goes into this bag. Okay.
So you'll go so air will collect in this particular bag. On this side, you'll get your cells, and over here you'll have a waste stream.
Okay.
So usually we get a waste in our particular process. It depends on your process. We will have maybe more than a liter of waste. So there's this bag over here. Alternatively, you can weld on another bag, which I've done here.
Okay.
Or you can weld on a bigger bag. As I said, I've checked all the clamps. I've checked the clamps on that side as well in terms of the stream. I have, as we had mentioned earlier, this would be my leukapheresis, my buffer, and my Ficoll. So the first thing I want to do is probably have some buffer run through the line to prime the line itself to ensure that when the cells do come through the line, they're not sticking to the plastic because everything in there is really precious. So right now this liquid will go down the path of the tube and the air inside the bowl will get pushed out and into the airbag.
Got it.
And then so usually you do that first, to just as you just mentioned, just to give it a prime the line itself. And then you can immediately close the clamp and then open up your cells in this case. Water with colored dyes just so that we can see.
And are these bubbles in the line problematic in terms of the cell viability?
Not in the beginning. So in addition to the line being primed with the initial buffer, you can, it also allows you to get rid of any bubbles.
Okay.
But at this stage, any bubbles that's in the machine is not too much of a problem. And you do have a bubble sensor. So later in the process of the machine, when you don't want bubbles, which is when the Ficoll comes in, and the reason you don't want bubbles is Ficoll is basically separating the layers of cells. You don't want a bubble to come in there and disrupt that separation.
Okay, because then you wouldn't have separation anymore.
Because the cells will end up getting mixed.
Okay. So the cell product that I want, right now I've got a mixture of cells, but what I want to get out of it is a certain cell type. So that cell mixture is flowing through here,
yes,
then going around the peristaltic pump, and then back through this valve, which is open at the moment,
correct,
and then into the centrifuge.
Okay. And because the cells are under centrifugation, they'll all pellet to the bottom, and any liquid would move to the top.
Okay.
Which in this case, in our case, would be all your red blood cells, including your cells of interest, your PBMCs, your white blood cells. They'll all be at the base, and any liquid that's in there that the cells are in, the plasma that you might not want in our process or in a Ficoll process, will get pushed up. So at this point, it's now detected that my layer of plasma is done. So it's asking me to move on to the next step. So I've now opened up the blue clamp, which is asking me to do, which will now collect my cells.
Okay.
So what I will do is I've collected all my cells, they've gone through the system at this stage, and then I'll open the Ficoll line. So this line's closed. Ficoll line, I'll clamp this first. Ficoll line, and then I'll continue the process. Press play.
Okay, so now I see the Ficoll is dripping in and chasing the cell sample through.
Yeah, because the Ficoll is much denser than the cells that are in there, the Ficoll should be pushing the cells down the system. And what will happen is in the chamber, under centrifugation, all your cells is at the base. And as the Ficoll, which is much denser, comes through, it will push your cells, your T cells, your B cells, your monocytes up, while your red blood cells, which are quite heavy, and your granulocytes, they'll tend to stay at the bottom.
Okay. So now as we're waiting for the Ficoll to push its way through, you've already opened up the blue valve, which is the cell collection bag.
Correct.
How does only the cells that I want come into that bag, and the cells that I don't want go into the waste bag?
So what will happen is, as the Ficoll is pushing the layer of your cells of interest up, it will keep moving until and eventually the red blood, so it will keep moving up, and it will the sensor here will detect that there's a change in contrast.
Okay.
So you've gone from a buffer to a layer that usually what tends to happen is it concentrates all your cells of interest to this very concentrated white band,
okay,
which is why it's called the white blood cells. And as soon as it detects that band, that's when the system will then shut down and ask you to close the clamp, which has your cells of interest, and open the waste stream. Since, as I mentioned earlier, this machine is not being used specifically for what it's built, but we've modified it based on an academic paper that came out to perform Ficoll. It's more hands-on and not very automated. So you have to be at the machine most of the time, and it takes about an hour to do the entire process. Unlike some other machines that might automatically open and close clamps, you physically have to do it.
So now that this process has gone to completion, I take my sample bag, and then what do I do next with my white blood cells of interest?
Before you started the process, you usually take a sample to see how many cells you have, so a pre-count, and then you want to see at the end how many cells I got back, and it gives you a little bit of flexibility in understanding. I had so many cells, how many cells did I end up with, and how much did I lose along the way, which should not, most of the cells that you've lost should not be the cells that you were interested in. In this particular case, what's going to happen next is you're going to have a little bit of Ficoll would probably have transferred over into your cells. And as I mentioned earlier, you might have a little bit of contamination of some of the other cells. So from here, the next phase would be to wash that cells.